Hi, my name is Aarushi Gandhi. I'm another technician in the Mussolino lab, and I started in the lab as an intern in 2022, and I was excited to continue my tenure in the lab as a technician for another year or so before starting medical school. And so today I'll be presenting my results on the effects of gene therapy on the blood-brain barrier. And so the blood-brain barrier, or the neurovascular unit, is comprised of three main cellular components: the endothelial cells, the astrocytes, and the pericytes. And so the endothelial cells in pink are made up of the first line of defense, and tight junction proteins maintain the integrity of this barrier. The pericytes in orange are crucial to characterizing the blood-brain barrier in MSMDS because they express SMA that the ACTA2 mutation is present in and contribute to the maintenance of the capillary blood flow. And then the astrocytes in purple are glial cells that are activated during neuroinflammatory events, and their astrocytic end feet play a key role in keeping the blood-brain barrier protected. And then the other relevant cells in the brain include the microglia, which are the brain's immune cells, and then the neurons. And so think of the blood-brain barrier as like the customs or the TSA at an airport. So they're what protect the brain from the different pathogens that are not supposed to enter the brain. And so we're looking at how these cells are treated and remain normal after treatment. And so on the left are the arteries from a human ACTA2 patient and a control. And then the middle cerebral artery, or MCA, possess a postmortem wrinkle artifact that is expected during processing. However, in the mutant vessels, they're so deficient in contractility because of the proliferation of the intima that they're unable to react to the fixation artifact that we see in the control. And then we see a similar thing in the ACTA2 R179H mouse, where the same loss of SMC contractility and wrinkle is recapitulated in these mouse models, which is denoted in the right with H&E and then SMA staining. And then so additionally, we were interested to see the integrity of the blood-brain barrier in these mutant mice. And so we did so in two different ways. We employed the two-photon microscopy technique to visualize the leakage of 10 kilodalton fluorescent dextrans over an hour in the mutants. And in this live mouse imaging, we revealed that there were extensive leaks over time. And then on the right is Evans blue injections that also confirm the same thing. And so in this ex vivo technique, the Evans blue binds to albumin in the blood, and which autofluoresces. And so we visualize these leaks through measuring the mean intensity in histopathological processing that we saw the same thing in. And so to correct the mutant, base editing of the ACTA2 R179H allele strategy was employed using an A to G base editor that can directly reverse the nucleotide change that causes the mutation. And so the base editing efficiency was studied in the brain vasculature, and the phenotype rescue was measured. And so this is the effect of the gene therapy. It was exciting because we were able to restore the wrinkle in both the large and small vessels of the brain. And by using AAVPR and AAV9, the vessels were able to contract again. And so what does this mean for the brain? The brain relies heavily on neurovascular coupling and a working blood-brain barrier to develop fully and not be dysfunctional. And so the astrocytes in the brain that were stained with GFAP, or glial fibrillary acidic protein, are very activated in the untreated mouse. And so the gene correction with AAVPR and AAV9 restores the normal astrocytic morphology in the metabolically active brain regions, such as the hippocampus and the motor cortex. And so the inflammatory brains of the brain, or the microglia, can also be seen in between the control and the mutant, that they're activated. And so the treatment with AAVPR and AAV9 seems to diminish the neuroinflammation in the brain and bring it back to control levels. And so this is a histological correlation of the malic basic protein, or MBP, in the brain. And this is where the white matter injury is seen in humans. And so the white matter injury is stained with MBP. In the mutant brain, there's less staining, which means that there's less MBP protein. And in comparison to the mutants that are treated, the restoration of MBP because of the gene therapy can be seen. And so in conclusion, the ACTA2 R179H mouse model recapitulates the blood-brain barrier and the larger vessel of human pathology. The vascular and blood-brain barrier dysfunction in the mutant mice correlates to the white matter and neuronal degeneration. Targeted gene correction by vascular-tropic AAV delivery of CRISPR-Cas9 adenine base editors restores the SMA expression and SMC and pericyte function. And the improved blood-brain barrier and vascular integrity ameliorated astroglia and microglia activation and also neurodegeneration. And so I'd like to take a moment to really express my gratitude towards the patients and their families for their time, and then towards Dr. Mussolino for her continued guidance and support, Dr. Lindsay, Dr. Krishnan, and the other scientists in the lab for their expertise in experimental planning, and the extensive network of collaborators without whom the study would not have come into fruition. Thank you.